Well, good morning. It's really a privilege to be here to get to talk about things that we're excited about. And you might see the word up there on the screen and say research. How could that possibly be interesting? Well, because I'm the first person up here, I suppose I better start by offering a little definition of what research is. And so here's a highfalutin sounding thing. Building the body of knowledge through systematic, purposeful inquiry. Now that you're on the edges of your seat with excitement, let me address something about that. When I was an undergraduate, I think building a body of knowledge was the farthest thing from my mind. I was much more concerned about acquiring the body of knowledge for the next test that's coming on Wednesday than I was ever thinking about this. But I have to tell you that as undergraduates, you're still inquisitive, and there are some pretty neat things about research. So why in the world would anybody ever want to do research, you may ask? And I'm going to offer three ideas here in the context of my own work. I think, as Dr. Curry just mentioned, we are curious. We are created in God's image with the ability to think and to ask questions, and we want to know about ourselves and the universe in which we live. We're curious. And then another really neat thing is that God gives us passions. Things that we're excited about, things that we just are driven by, and those passions will oftentimes fuel questions that lead us to want to do research. And then third on the list, and by no means the least, is this idea of need. We don't know enough to handle all the problems that we face in the world, and some of those problems are external to us, and some of those problems are internal to us, and we can be very much motivated to do research by that as well. And I'm going to submit to you that all three of these are extremely important to me as I do the work that I do, making pictures like this using something called real-time magnetic resonance imaging. This is looking at structures inside the body, not as static images, as you typically might see in magnetic resonance imaging, but rather at speeds of 100 frames per second as movies, looking at movement. It's a really fascinating area. And my application is really esoteric. I'm interested in looking at this movement in the context of studying brass performance, wind instrument players. And I get to do this here. This is Göttingen, Germany. This is the Max Planck Institute for Biophysical Chemistry, just outside of Göttingen, the city of science. And I spend my time there collecting data and bring it back here to work on. It's a great place to go. Before I talk about the research itself, I want to talk a little bit about the background that brought me to this. I think it's important. And I've got a real busy slide here with lots of information. I'm just going to whip through real quickly. I was born into a musical family. My mother, my father, my uncle, all were French horn players. My grandparents were professors of music at the University of Wisconsin. And in fact, there's a picture of my dad playing principal horn, my mom playing second horn, my uncle on third horn in the University of Wisconsin yearbook back in whatever that year was. All of them were musicians, and there was very little chance that I was going to walk away from this family not being a horn player. So when I graduated from high school in 1972, I started out as a French horn performance major at Indiana. I had a really hard time. I was very much a perfectionist, and I had a case of performance anxiety that, quite frankly, made it miserable for me to play and make music. It took all the joy away, and that passion I threw away. I just stopped playing completely, and I turned my attention to one other passion that I had, and that has to do with human movement and how we exercise and train. So, to make a long story short, I ended up pursuing a PhD in exercise physiology at the University of Kansas. 28 miles away in Topeka, Kansas, my father was teaching in a music program at Washburn University and also playing principal French horn in the Topeka Symphony. And he said, "Pete, get your horn out. You need to audition." And I spent years with my father during my PhD actually playing side by side with him some of the greatest literature the orchestral world has to offer. It was a great thrill for me. But the pressure was gone, so it was a different experience. Well, I came to Gordon College in 1983 as an exercise physiologist, but I kept my horn in my back pocket, and as the years went on, I got it out and was encouraged to use it quite a bit, and as you can see, I had some experiences as a professional horn player. That is until 2001. In 2001, something happened. I started feeling something in my face when I was playing that told me something wasn't quite right. And I sought the advice of world-known neurologist. This is Dr. Stephen Frucht. He's in New York City. He's an expert in movement disorders in musicians. And here's what he told me. He said, "You're done. Put your horn away, find something else to do. You've got embouchure dystonia. Now, I have to tell you, I'd started playing when I was 8 years old, and to get that news was crushing to me. It was an identity crisis. It was really hard. Well, what is it? Let's talk about that just for a minute. Dystonia is a class of movement disorders where you basically can't control your muscles. They spasm or they have too much activity. Musicians have a very special kind of dystonia, though. For us, the things that are affected are those movement patterns that are the ones we've trained ourselves most to use. Pianists lose the ability to play with their right hands. Violinists might lose the ability to move on the fingerboard. For me, it's the muscles controlling the mouth no longer working. I can't buzz right anymore. Let me show you a little bit of what I mean by that. So listen and watch. Unsettled. What you no doubt can hear is a very shaky sound. Let me tell you, you don't get orchestra gigs sounding like that playing on a mouthpiece. But you also, if you look carefully, could see lots of quivering going on in my facial muscles. And no doubt you've noticed there's an electrode sitting there on my face measuring muscle activity, because I was really interested in this. When I got the disorder, I wanted to know everything that I could about it. I had to take stock of the situation. Lots of people were studying dystonia, but I had sort of a unique approach to it. The approach is that I had the disorder, I'm also a physiologist, and I'm a horn player. And that set of gifts, if you want to call it that, including, I'm going to say, the gift of dystonia, made for a very, very providential opportunity that has been extremely exciting, and I want to tell you about a little bit. Early on, I had simple questions. For example, when I played, my face got all wrinkled up like this. And if you saw my brow, which you can't see in this picture, it's all furrowed. There's all kinds of extra muscle activity that has nothing to do with buzzing on that mouthpiece. And when you compare that kind of a facial expression to one of my poor students, oh, they got electroded to death, I must tell you. But this is Mindy Hofsass from the 90s. Her face is extremely quiet, and her embouchure is doing just what it's supposed to do. And so I had this idea, can we use biofeedback so that I can retrain my nervous system to shut off the extrafacial nerve activity that's leading to that tension? And I did some experiments, and I wrote about this, and was fortunate enough to get it published in a journal called Medical Problems of Performing Artists. And I was invited to speak at the Performing Arts Medical Association in Aspen, Colorado in 2005. It was a really great opportunity to share what I was doing. Little did I know that in the crowd was a very important person. This is him. His name is Dr. Eckhard Altenmüller. He's the director of the Institute for Music Physiology and Musicians Medicine in Hanover, Germany. The building you see there is where his offices are. He has the entire upper 2 floors of that building. And he said to me, he was very interested in the research, and he said, you know, sometime you should come to Germany and we should visit and you should work with me a little bit. Well, I wasn't too bold to do that, but then I had an opportunity in 2012 to put in a sabbatical request. And I wrote to Eckhart and I said, do you think there's anything that you might be doing that I'd be interested in? And he said, oh yes. Right now we're using MRI to study movements inside the mouth of horn players with embouchure dystonia. Perfect time to come. And so off I went, sabbatical of 2013, jumped on a Lufthansa flight, and off I went for a whole semester of work there. I have to tell you, I was petrified. I had absolutely no idea what I was going to be doing. I just knew there was something exciting there, and what was it going to be? So I got there, and the first thing he does, sits me down in his office, and he tosses me this hard drive, and he says, okay, Peter, here's the data. All these films of people buzzing on a plastic mouthpiece inside the MRI chamber. What do we do with these data? Let us know. Great. So for 2 months, I addressed a couple of questions that came to me as I was looking at these films. Films are nice, they're descriptive, you can see things happening, and you'll get to see one in a minute. But I was interested in getting quantitative data from these films. How could I do that? So we worked on finding a quantitative way to measure things and developed a new testing protocol that involved the development of some interesting things that I'll move through quickly here. These are my collaborators at the Max Planck Institute. This is the director of the facility, Jens Fromm, and some of his associates. These guys are brilliant scientists who have the technology that needs a place to be applied, and they were very eager for me to use it. And I had to give a presentation to these guys in Göttingen. It was really intimidating, But one of the ideas we came up with was to develop an MRI-compatible French horn. This is Dr. Richard Serafinoff from Indiana University. He built us one. The bell of this horn goes outside the magnetic field, and the hose runs in so the person can play it inside the chamber. And voila, we have a real French horn that feels like a horn, sounds like a horn, and mimics playing much better than playing on a plastic mouthpiece. And then I got really bold. I said, we need to study the elite players of the world. So I took my little bag and I took the train to Berlin, and I started recruiting the Berlin Philharmonic French horn section to be my elite subjects. The idea being, if we can study elite players and learn from them, maybe we can find things that are common to great players that could help our dystonic players, perhaps. Maybe there's some differences there. The data looks like this. I'm going to quickly play this for you. Essentially, what you're hearing is this sequence of notes being played. We impose a grid system inside the mouth, and if you watched carefully, the tongue was moving along these lines. From those data, we were able to generate two-dimensional graphs here that you can actually derive quantitative information from. So we used this technique to get lots of measurements, and what we found was that all of our elite players, when they play up to high notes, we get this pattern showing up in their graphs, particularly in this graph right here, a rising pattern. If we look at the physics of that, it makes perfect sense. What they're doing is they're making their airway narrower, and it's helping them to play the higher notes more efficiently. This was really exciting. And it was common to all of our elite players and not there in our dystonic players. Well, that's the kind of data we did. Where has it all led? I want to just quickly talk about that for a minute. One thing that happened was Deutsche Welle Television got wind of this. The woman sitting right here in the middle is Sarah Willis of the Berlin Philharmonic. She was the first woman ever to make it in the brass section of that orchestra. And she was so interested, she brought her own television show to our Max Planck Institute, and we filmed a series called Music and Science, and you can watch it. It's April 2015 if you want to go back and see it. Fascinating stuff. And then she took her videos of herself, put them online, and within 3 months we had 42,000 hits. People fascinated by seeing these things. And we, in turn, a colleague of mine, Eli Epstein, who was one of our subjects, he's a teacher at Boston Conservatory and at New England Conservatory. And he and I teamed up to give a presentation at the International Horn Symposium in Los Angeles back in August of last year. Me talking about the science, him talking about the teaching or pedagogy that comes from this. So in a very applied sense, the research we were doing was helping people learn how to play the horn better. And we're right now working on a website that has a series of lectures and videos that Eli and I are putting together to inform the French horn and brass world. But that's not all you do when you do research. You don't just get popular things out there, you write papers. And so we've been very blessed in the last 2 years to come out with 4 papers dealing with the research that we're doing, and it's getting a lot of attention and interest. And the last one at the bottom here involves Rachel as my assistant last summer. So I'm going to have her talking about that in just a minute. But what I want to finish with is this. It's a passage from Corinthians, 2 Corinthians, that I really own. I don't need to read it. You can read it. God has turned this disability into an opportunity for me, and it is really a wonderful opportunity. So thank you very much. Rachel?
Thank you, Dr. Iltis. Thanks for giving me a chance to share. My name is Rachel Burke. I've been working with Dr. Iltis on this research since my sophomore year of college. He approached me in that fall, asked me if I wanted to be involved in his research, and at the time I knew absolutely nothing about French horn playing, nothing about musicians' focal dystonia, and very little about the technology used to analyze these films. I did have one connection though. I've played violin for about 17 years, so I understood the musicians aspect of it. As a kinesiology major, I'm also very interested in the science behind a movement disorder and the development as well as the production of this movement disorder. I grew more familiar with the research over this semester, and when the opportunity came to apply for the Provost Summer Research Fellowship this past summer, both Dr. Iltis and I were very, very excited. We had one particular research question. He mentioned it briefly, but it's asking whether the elite players and the dystonic players show a distinct difference in movement strategy We were particularly looking at ascending and descending harmonic sequences. It's basically a scale, a set of notes that increase in pitch and then decrease in pitch. We were looking at patterns of mid-tongue and jaw displacement. Very particular question, but it's relevant for those with embouchure dystonia, perhaps. We did see great results. We saw differing motor strategies for the tongue and jaw on ascending and descending harmonics. As you can see in this graph, it shows mean jaw movement for descending harmonics in particular. I want to point out that the black bars represent the elite performers. Their movement pattern shows a very distinct drop in jaw movement as the notes decrease in pitch. The dystonic players, on the other hand, with the white bars, show a less consistent pattern, and the magnitude of change is also a lot less consistent. So, we were thinking that the less efficient strategies of dystonic players may be a contributing factor to embouchure dystonia, and that's a really exciting finding. It's opened up a lot more questions, and we're pursuing vocal fold patterns of movement. That's kind of a next area of research that we're working on. So apart from getting results, why research? And this brings me back to how Dr. Iltis opened. It comes down to curiosity. Even though I knew nothing about the project to begin with, I'm curious about learning things. So I'm really thankful that I got a chance to work on the Provo Summer Research Fellowship for that reason. But as a result of the summer, I learned a couple things about myself. I really enjoy working in a lab for 8 hours a day, focusing my mind and my attention on one particular project, problem solving, creativity, and I was given a lot of freedom to pursue those areas during the summer. There's this quote, it's attributed to Einstein, he probably didn't say it, but it says, if we knew what it was we were doing, it would not be called research, would it? That's how we started, but as a result of the research, we have a lot more questions, and we do know now a little bit more about what we're doing. It's opened up a whole range of opportunities and really moving forward on the project. So I'm really thankful to have been a part of it. I think that as a result of being involved in research, I would be okay with pursuing it as part of my career. That's an interesting thing I would never have considered before these last couple years. So thank you for giving me a chance to share. I think Dr. Keller has a few things to share about his research. Thank you very much.
Good morning. Seriously, my talk today is going to be participatory, so let's join. Hey, good morning. Oh, that's lovely. Thank you so much. My name is Greg Keller. I know a lot of you, and it's wonderful to see all of you here today. I am in the biology department, and I work in landscape ecology and conservation biology, and I work with lots of students, and today I'm going to share some of my work that I've been doing with Katherine Schweitzer and Lauren Purdy. We were really lucky and blessed and fortunate to be part of the summer fellowship program last year. So I want to share with you some of the things that we did and what we found. I'm challenged, though, in this talk. Unlike Dr. Iltis, who has an incredibly compelling story that he shared with you, I don't have that. That's miserable, isn't it? When you think about, hey, tell us what you do, and you think for a second, I don't really want to. I'd rather not, thank you. At some point in your lives, you're going to have that problem. You are going to be stuck at a dinner party. You're going to go with your spouse or your partner, and you're going to realize that you know no one and your spouse knows everyone, and so they quickly wander away, and you're stuck by yourself at the buffet table with nothing to do. And slowly but surely, you sort of look around, and you realize there's another person there. They're also sort of stuck with nobody else to talk to, and now I have to do something. And so you wander over, and you say, professor something, research, and I sigh deeply because I realize this is the start of our conversation that I don't really want to have happen. And next, you say with some level of fake excitement, yeah. And that's the downside, isn't it? Super something great. A professor. Stuck with the professor at a buffet table at a party where you know no one else. It's like a nightmare for every student or every partner. Well, my answer all can be honest, and I can choose the way in which I want to answer that question very carefully. I can say with great honesty, I study Amazonian rainforest migrants if I want to try and impress you. And perhaps I do. You're speechless and our conversation ends, and thankfully for maybe both of us, we're finished with that conversation. This is an honest answer, and perhaps you're thinking to yourself, he's the guy who goes on these cool expeditions to the deep Brazilian Amazon, and he studies indigenous peoples, and oh, how cool that is. And I'm thinking, I study birds that migrate to the tropics. Perhaps if I really want to frighten you, I say this in my answer. I study the genus Dendroica in Perulidae, for you see, I'm a scientist. And perhaps you might say in response, if you're an education major, because you realize, or I certainly realize, that the fastest way to end any conversation is to use Latin. Is that true? And so, in using the Latin name for warblers, Dendroica, I know exactly what I'm saying to you, and I have frightened you away. My plan is working perfectly, and I get back to the Swedish meatballs, giving you a sinister look. Again, I'm answering truthfully, and what I can say to you with great truthfulness is I study edges. In my mind, I think of this edge, this forest edge boundary where migratory birds might be concentrated. And of course, fine art students, you might say, because you think of the edge, not edge, when you think about what I might study. I might say to you, again, with great honesty, I go on expeditions in my work, and you think to yourself, if you're a woman perhaps, yeah. And I continue my sentence because you cut me off, to study things in people's backyards. Yeah. So what you've come to realize then perhaps is I study really perhaps interesting things in kind of strange places. I study things in backyards, at edges. I go on expeditions. That's my work, and that's what I'm passionate about. But trying to explain that to somebody, there's no clarity there. Perhaps the worst answer for me is this. And this is the answer that I know it has to come eventually, but I dread. Because as I think about what this conversation might look like, I study birds. I study birds. That's another conversation killer often, except for some of us. And perhaps if you're a biologist, you might say, because at every dinner party, if I say I study birds, everybody and their brother has a bird that they've seen somewhere, and they're convinced that I can identify it. They just say, oh, it's brown. You know that one? Here's the thing. I do know. Yeah, it's a yellow warbler. Of course I know that bird. But biologists, you know that's not the end of our conversation. With a bit of anger, you might say, yellow warbler. Trust me, people, I know what I'm talking about, all right? Well, that's not necessarily the end of the conversation, but it certainly hopefully shows my passion, my career direction, what I have done in research. Research is a passion of mine, just like with Dr. Iltis. It's passions that we have. And to have an experience like last summer when I can explore that passion deeply and broadly and try to understand something fully that's unexpected is really fascinating to me. So that's the value to me of the summer fellowship last year. But let's get a little bit deeper. Because if you're a good conversationalist, you know that these questions of what don't really help us very much. Professors? With feeling, please? Professors with, I see you sitting there. Professors with feeling, please. Thank you, Peter. I knew we should have had a departmental meeting before this. I knew I should have talked to you. I thought it would work. Okay. here's the thing that's maybe more meaningful. As we start to look at that passion that is birds for me and research along with that, why in the world? And that's the more interesting question, is why? You heard Dr. Iltis share, why study dystonia? That's a really deep question. And so if you're a good dinner party guest, that's your question, is why? Certainly, one of the major things is birds are often pollinators. In Mexico, 85% of agricultural crops require pollination. Think bananas and agave and papaya, the great fruits. They require pollination. Lose birds, lose bees, lose beetles. We've lost agricultural crops. Birds are significant. Maybe like me, you enjoy a latte or a cappuccino or an espresso or just a plain old cup of coffee. There's a wonderful study done in Jamaica. If you exclude birds from a coffee plantation, yield goes down 14% because of the coffee borer beetle. It's a little tiny larvae that eats coffee beans, and it degrades them entirely. Do you like coffee? Do you like coffee? Yes, of course you do. You're always in line in front of me at the bistro. Yeah. So yes, you love coffee. You know that you do. But birds control that coffee borer beetle. 14% greater yield with songbirds present on these farms. So buy shade bird coffee, buy shade grown coffee, buy organic coffee, because that's bird coffee. It's fantastic. Why else would I study these organisms? I mean, they're beautiful. Look at this black burning warbler. It's gorgeous. But importantly, these are in severe decline. About 1% per year decline for migratory songbirds. And so as we think about the roles they have in an ecosystem, they're critical members. And finally, as we look at this imposing edge of a forest, what I also realize is, although we've been studying biology, ecology specifically in our region for centuries, we still know very little about how we impact, through environmental degradation, how we impact biological communities. And so this is what I've been doing. This is my work. How do humans unintentionally affect biodiversity, especially in the form of birds and mammals? I look at different edge types. I look at residential edges, which is essentially truly your backyard. I'm probably not wandering your backyard without permission, but I do wander in people's backyards. I look at wetland edges. I'll show you these pictures in a moment, and pasture edges in fragmented systems. And then what I really want to know is, at a big scale, at a very broad scale, how do natural heterogeneity, like our ponds, like koi pond ecosystem, how does that compare to human-induced degradation in the form of fragmentation? So I look at habitat use of birds, I look at behaviors of birds, and I chose 8 focal species. Right here in Massachusetts, in northern Essex County, Katherine and Lauren and I went out and we're counting birds and trying to understand their behavior. What I was expecting wasn't necessarily what I found. I was just hoping for anything. If you're a researcher truly, you hope for anything. And I'll explain it later, but just find me something significant. So I thought, there might be greater resources at a residential edge. Maybe because you water your lawn, it might have more insects. Maybe natural communities, maybe pasture edges have more resources as well. Maybe they're more natural because they have some grasses growing. Or maybe those wetland edges, maybe they are less degraded, and so that might be it. So these are the types of habitats we were looking at. This is a wetland edge. This is essentially your backyard with a little fence and a paved, why would you have a paved walkway in your backyard? Why do you do that? And then also a pasture edge. So you see some natural vegetation growing here. It's still degraded, but that might be it. So I want to show you some results here because you always fear as a researcher, you come up with this fantastic project and it was accepted and I got wonderful donors, but are we going to find anything? And we analyzed our data that Katherine and Lauren brought back in, and I was gleeful when I saw these numbers. So let me show you what we're looking at here. I have 3 edge types: natural, that's natural heterogeneity, pasture edges, and residential edges. That's from left to right within the groupings, and then 4 different species that all of our 8 all showed significant trends. Really, actually, trends is not the right word, significant patterns. Look at the colors here, though. Look at the high bars. We're looking for the greatest abundance with the high bars, and look at the numbers here. By far, almost double, in some cases triple the number found in natural edges versus the others. I was truly gleeful because we worked hard. Katherine and Lauren were out every morning at 5 a.m. and it's hard work. We found something really fascinating though. Well, that wasn't the end of our project. So, great, natural edges, hooray. They are truly natural. They are not degraded. They do have more birds, and that's a fantastic thing. This is a common yellowthroat that you're looking at, by the way, a little tiny warbler. But what I really want to know is at a big scale, does big scale fragmentation affect birds, affect these 8 focal species? And so this is what we do. This is kind of cool. You can see this is an aerial image, a satellite image of our area here. And you can see a residential development on the top right. You can see some forest there in the green. You can see some wetlands, the little ponds that look black. I want to know, does something way over there affect something right here? Can we study that? And that's the technology that I use is called geographic information systems. It's a way to look at big scale things. So our study site here in the middle, you see that little disgusting green dot. We establish a 500 meter radius circle, and we simply say, do birds respond at 500 meters? Essentially a little more than 5 football fields away, do birds respond to those things? So we delineate wetland edges, that little tiny blue squiggle there, that's wetland edge. We delineate human edges, those are the disgusting yellowish color there. And you can see this is a highly degraded system. This is a highly fragmented system. And then we also measure the amount of forest within this 500 meters. This is landscape ecology at its finest. And what we found then was, as we analyzed our data, do birds respond to big, giant scales? Thank goodness, yes. Great crested flycatchers on the left, top left, warbling vireo on the top right, and Baltimore oriole, you all know that bird, on the bottom middle. These are all birds that had a significant response to fragmentation. The more fragmentation at a big scale, the rarer these birds were. We were stunned. Birds respond at giant scales. Now, not all of them. These 3 had no effect. This is a yellow warbler on the top left, a common yellowthroat top right, and an eastern kingbird. These birds had no response. So they responded to edges, but not to edges. And then finally, strangely, there were 2 others here, red-eyed vireo on the top right, top left, and gray capper. These are ubiquitous. These are ultra-common species, and they didn't respond to fragmentation. Actually, they did. They actually occurred more positively with fragmentation for some reason. So, great. Birds respond. Hooray, we chose the right group, we chose the right species, we chose the right study. Thank goodness we were funded, we could do it, and we thought that was the end of it. But then we started doing something else. Okay, so a bird is present. Fantastic. But is it breeding differently based upon these patterns? From the balcony, are you surprised? So one of you is not texting. Thank you. That's wonderful. So I want to walk you through this because this was incredibly surprising to us, and we had just no idea we would find this. We tried and we thought maybe. This is, again, the common yellowthroat. And so what Katherine and Lauren did was go out, and they didn't just count birds, but they also watched their behavior. And they said essentially, does some measure of behavior differ for a given bird species? And they simply counted bird songs. And so you saw that yellow warbler singing a little while ago. And they simply counted over the course of 3 minutes, how many times does a bird sing? It's kind of boring. I'll grant you that. It's not super exciting to actually do it. But to find that pattern was amazing to us, that we found that bird breeding behavior differed based upon the amount of natural edge. So what this graph shows, for every bird that we followed, how many times did it sing? That's our y-axis. And how much natural edge was there in this big 500-meter radius circle? So you can see in some places there was a huge amount of natural edge, and birds sang more. What in the world? Why? Why would a bird sing more in a landscape with more natural edge? We were stunned and excited, and we're still trying to explain that, but it appears that maybe birds sing more if it's a higher quality male, if it's a higher quality territory. And so the more natural edge at a landscape scale, the better quality the territory is, which was really fantastic. Now, I didn't do this every day. I certainly didn't do it alone. And so I want Katherine to share with you some of her ideas about what this summer was for her, how this summer looked and what she did, but more importantly, where does passion come into this question? So as we invite Katherine, would you please, Spanish-speaking people? Seriously, nobody here speaks Spanish? Okay, very good. Katherine, let's hear it.
Hi, I'm Katherine Schweitzer, and I love birds. Anybody and everybody who's ever met me will tell you the exact same thing. I grew up on a bird farm, a hobby bird farm, where we had pigeons and chickens and turkeys and ducks and pheasants and canaries and doves and everything. We had it, and then we had some more. I've been a bird watcher since the first grade, when my grandpa taught me how to tell apart a black-capped chickadee from a dark-eyed junco. And I can remember the exact day when I could tell my mom, "Look, that's a dark-eyed junco." And she's like, "That's nice, honey." I have tomorrow a 12-hour birding competition with Dr. Keller, where we're gonna go out at 5 a.m., well, leave earlier than 5 a.m. to get there, and count as many bird species as possible from 5 a.m. to 5 p.m., just because I can. I have story after story after story of me either making a fool of myself and being really weird about birds, or friends coming to me with a question that they have about a bird they saw, or just saying, "Oh, Katherine, I saw a bird and when I was walking and it was really cool and it was singing and I was walking along the path. What was it?" The same thing. It's the same questions over and over, but I love it when my interests make my friends want to learn more, and I'm always super happy to share with them about birds. So I reiterate the fact that I really, really love birds. I'm a bird nerd. And more often than not, the response to my enthusiasm is something along the lines of, "Katherine, oh, you and your birds, that's so fun, that's so cool that you like them, oh, they're so sweet." And I completely understand, because I grew up around them. I grew up on a farm and surrounded by them, and my parents were both really encouraging to me in that, and that's not the case for everyone. You didn't have 400 pigeons. I understand. I can't imagine not loving them as much as I do, but I understand that not everyone does. So all this is to say, I was going to do bird research with Keller regardless of the Provost Summer Scholarship. I was hoping and planning to do it this year, my senior year, and the fellowship was more of a perk. I was like, "Oh, I can earn some money doing this. I can put it on my resume. This will be a great experience." But honestly, I didn't think that the project was going to get accepted. Because, Keller is an ornithologist. He studies birds. That is what he does. Of course he's going to want to pursue this and be interested in it. And of course I'm going to want to, because of all of the reasons I said. But other people aren't as excited about birds. And in my experience, I've had to really tone down my enthusiasm around when I talk to other people, because I can get pretty intense, it's crazy. And so I didn't think that it would be accepted. Why would somebody want to pay me and want me to study birds? You don't like birds, so why would you want me to do that? And yet it was accepted, and it's crazy to think about that other people care about what I care about and what I'm passionate about, and it matters to someone. So I didn't make any of those connections over the summer, and it's taken me a long time to think about it, and the more I've thought about it, about why I did the summer research and why it was important, why it mattered, it's become more clear, and that, yes, it's important for all the reasons that Dr. Keller mentioned, where we're learning more about the effects of habitat fragmentation and the really important vital repercussions that have on species that live there, and in ways that we're only just beginning to understand. And I can really easily say something along the lines of how this was a great resume builder and a great learning experience, and I learned so much about my field of biology and new techniques, because all of that is very true. But I don't want to say to you, go do research because it's the best thing ever and everyone should do it, yay. Because that isn't my message and that isn't right. After the summer, I really realized that as much as I loved doing research, it was fantastic, I might not end up doing research in the future. Rachel found that after she did it, she was like, "Yes, I could see myself doing this. This is wonderful." And I did it, and I was like, "This is great." But it's not the only thing for me. So it's completely different. And I'm guessing that that's the case for a lot of you. For most of you, you're not going to go out and do research. But I'm also going to take a guess and say that you have something you're passionate about. I'm passionate about birds. There are people who are passionate about dystonia. Maybe you're passionate about history or chemistry or physics or math. Oh goodness, math. About theater or art or Christian ministries, something that you are passionate about. And it might be something super specific, like birds for me. And it might be something that other people don't get. They don't understand why you would do that. But don't let the fact that other people aren't as passionate as you are about something stop you from doing it and pursuing that. Study things not because it's considered a normal field or typical, but because it's something that inspires your curiosity and it captures your passion. So you can do research or don't, but as long as you do what you're loving to do, then there are people and there will be people around you who support you in that, like the provost in the summer research I did. So yeah, I'm sure that if you do pursue a field that fills your passion, then it will also inspire others to do likewise.
Well, as we finish up today, let me share with you some overarching lessons. First of all, from our research together, it appears that there are subtle and unexpected things to find that are sometimes curiously contradictory. But that's what really makes research fascinating, because we continue to ask those questions. Biodiversity and behavior are both impacted negatively by fragmentation. That is a giant point for us to understand. The natural heterogeneity at a big scale really does affect things, and we need to focus on that a bit more. TSC students, say a word here, please. 2 words. TSC students. But why? But why? And that's the big point for us next. Why? Why in the world are these patterns true? So for Dr. Iltis and Rachel and Katherine, for others who have done summer research, and for those of you who might be applying in the future, thank you. Thank you to the college and the donors, because truly it changed our lives this summer, and it was a wonderful, wonderful experience. For you, keep asking why questions for your dinner parties. They're far more compelling. You, with sincerity, have a wonderful day. Thank you very much.